recorded for first in the morning session. As John started his talk trying to describe the Asian Eye Genetics Consortium, trying to say that virtually this is the fourth meeting that we are having in a sequence. It all started in Hyderabad when John tried to reach at me and wanted to have all the eye genetics group to be collected in the for one of the AOC meeting. At that point, when I searched across India, most of you here would be surprised that there were about a dozen groups right from northern India, Punjab and Haryana till Kerala. And the most active group, in fact, was that of three institutions, basically Dr. Sundaresan at Arvind, Shubho at LV Prasad, and me and my group at Shankar Netralaya. So we all contributed quite significantly, but I'll show basics because this is an ophthalmic crowd, and then a bit of our experience and the current status, as I told you, about eye genetics in India. There are about roughly 600 genes that are ocular in nature, one of the highest in terms of a single organ in the body. And this is not totally 547 because some of these disorders could cause, some of the genes could cause both anterior and posterior segmental anomalies, and therefore might contribute in different segments of the eye despite being one gene. But as we told you, ophthalmology and genetics is a big component in the entire perspective of human genetics. Those are some of the genes that you could see right from the cornea till the optic nerve. That causes a variety of disorders, and if you include the lid and the eye as such, then as I told you, there are about 600 different genes. Since my talk is on retinal degeneration, there are about really 200 genes, a third of the genes of the ocular in nature. This is simply because the anatomy, physiology, and biochemistry of retina is one of the most complicated that you can ever think of in the entire human body. I'm not going to go into the details, but it's highly complex, and when you do genetic counseling to these patients, it's sort of a nightmare trying to make them understand, and if they are looking at the future family, then it's much more complex. But all that is now possible, which we couldn't do about 2.5 decades ago so precisely. So why is it important that you guys do all kinds of genetics and find out 600 different genes is the biggest question any clinician would have in their mind. But as you could see that in the eye with retina, and then you have the retinal pigment epithelium and the choroidal vessels, this is where the entire dynamics of all these genes play sort of a group activity. It's like a symphony. They all work so perfectly well, and there are two different mechanisms. One is what you see here, which is in the retinal pigment epithelium, which is the visual cycle, and the second one is the phototransduction pathway. So all these genes work together, and then when I'll show that a bit better way. When the photon strikes the rhodopsin molecule, that triggers the phototransduction, and then that ultimately results in the signal being produced that goes through the optic nerve to the occipital lobe for us to understand that as vision. But the role of the RP layer is much more sort of a regenerative potential, where it sort of gets all the molecules that have been used up and sort of stocks that up once again in the neuroretina, so that you can have a constant phototransduction that happens. Understanding all these genes gives a lot of importance in genetic counseling, and I'll tell you some examples how that happened. Let me tell our experience as such, and if you see that for about 90 different publications, a quarter of that that I was involved with involves the retina. So these are some of the significant publications, and of course these are different groups in which I worked in New Zealand, US, as well as in India, particularly Shankanetravyad. So it's a long way that I came across, and then I was groomed by two different gurus of mine. One is Dr. Michael Denton, who you can see on the left-hand side. I worked with them in New Zealand. And then, of course, Fielding Hitchman Sick, who's at the National Eye Institute. And my stay at the National Institute gave me a lot of knowledge and experience of understanding the whole field and getting the right contacts and the network that I'm currently doing. So this is my stay, and you could see Dr. Sundaresan also here with me. And my stay with Fielding, as I told you, was one of the most eye-opening part, and that still sort of survives and connects. And my connection with John Prakash is because of that. Of course, this is a time off that we took as part of our AGC activity. It's Shenandoah National Park in the US, where John was kind enough to take, and his wife really made a sumptuous lunch for us, and we all enjoyed. So that's Fielding, Nutty, John, and myself. The potential of my part of the world, particularly southern India, is consanguineous families, and that's how we, as Shubho said, we had a good pedigree, and then we worked with her, along with Dr. L. Vijaya, who's in the audience. She was leading that program on glaucoma prevalence as well as genetic study. So this potential of ours is a big strength. In fact, I was talking with Professor Iwata this morning in breakfast about this potential that we have, which we can still utilize in the Asian Eye Genetics Consortium. So this is a family with foveal hyperplasia. The father had it, and two of his daughters also had. So anybody coming from a Caucasian or a Western population would straight away say that it is dominant, but whereas this was actually autosomal recessive, this is called a pseudodominance, where it might look like dominant, where you can see in the parent as well as in the child, but it's a recessive disease, and because of highly consanguineous marriages, you could see one here, another one here. The recessive appears to be a dominant. Now that's the fundus image. I'm not an ophthalmologist, which had foveal hyperplasia. And we studied this family, did homozygosity mapping, and the gene was SLC38A8, which is sort of a neurotransmitter in the retina and producing this effect of foveal hyperplasia in the patient. First, it was a collaborative study which we did with Chris Ingelheim's group in Leeds, and then they had few families from Europe. And this is a collective mutation profile of various families that were isolated, and one of that was our family, which was from Narayanetralia in Bangalore, where I'm right now working as well. So we did a Marfilino study, and somebody wants to know a bit of this. I can tell you more detail. But this was done by another collaborative group of Chris Ingelheim. And what we were trying to see is that this was not involved in the melanin pigment pathway, and therefore we also checked with the patients whether they have any kind of pigmentation that was there either in the ocular cutaneous region, and we didn't find that. Okay, so therefore clinical involvement in any kind of research project is very much necessary. So we had to go back to our clinicians and ask them if our patients had any of these features. Of course, this was published in the American Journal of Human Genetics a couple of years ago. So what is that status that I'm talking about, which is still sort of depressing, and why we really need Asian Eye Genetics Consortium is what I'm going to talk to you about. So this was a slide that I prepared for a meeting in Colombo, which was a SAARC nation meeting. And what you see is these are all the SAARC nations, right from Afghanistan till Sri Lanka. And I went into the PubMed and put certain search words, and then found out that how many publications that we can sort of retrieve from the PubMed database. And what you can find, I'll just show you only the sort of total, okay, of work being done. So these are the total of all these things, which is retinitis pigmentosa, congenital cataract, diabetic retinopathy, glaucoma, genetics, that has been done across the SAARC nations. And you could see that in India there are about 250 publications that could be retrieved. This was not an ideal way of searching and getting information, because these are very loose terminologies, and therefore it might not be an accurate data, but it is fairly reliable information. And you could see any country in the SAARC nation is right on top is India. And the closest is actually Pakistan, where I could see that there were totally about 47 publications that could be retrieved. And the remaining countries were virtually nowhere near. And some of these were clinical publications that I could see in Bangladesh, for example. Now, if you compare that with US, okay, you can see why it is depressing. It's 6,247 publications that are there, and we were not even anywhere close, not even sort of 10% of what is being done in US. So therefore there's a need for ophthalmologists as well as basic scientists in India to work together, and then we can sort of scale it up to the Asian level, and then to the global level as well, as John Prakash mentioned. And now, okay, fine, you guys are doing all these kind of work. So where is this taking us as clinicians, and as taking up with another normal child? Okay, so this is the expectation with which we see patients in India right now. So this is the patient I saw in Aditya Jyot. Dr. Natarajan is the head of that institution, where I saw a patient which was clinical diagnosed as RP, and then when we did a mutation screening, the ABCA4 gene mutation was identified. There are reports of RP patients with ABCA4 mutation. Okay, very few publications are available. So the point I'm trying to make here is that we, as lab people, could help the clinician in trying to make a diagnosis when there is clinical ambiguity. So this is possible now, and I sort of use a company called MidGenome for this, generating that data and doing genetic counseling. So we basically outsource it. So with that, I come to the end of my talk. So I did tell about the basics and our work, and then about current status of we have to do a lot of work to be done. And I've been in this field of eye genetics, and also particularly in retinal degeneration, for about 2.5 decades. So still have not understood it. It's much more complex than it was taught in terms of in a clinical situation. So and therefore jointly along with John, Nati, and Professor Iwata here, I like to say that we have to form a good group in India and take things forward. And with that, I would like to thank all of you.